I'm Joe Graydon.
And I'm Terry Graydon. Welcome to this podcast of the People's Pharmacy.
You can find previous podcasts and more information on a range of health topics at peoplespharmacy.com. The FDA has granted emergency use authorization for 2 vaccines. How will they change the trajectory of the pandemic? This is the People's Pharmacy with Terry and Joe Graydon.
Today we're talking with the coronavirus hunter. He's been studying coronaviruses for 3 decades. He has unique insights on the impact of the vaccines on COVID-19. How soon can we expect to see significant progress?
What have we learned about the SARS-CoV-2 virus? Are we better prepared for the next emerging pathogen that could cause a pandemic? Dr. Ralph Barrick will share his expertise.
What recommendations does he have for safe family gatherings?
Coming up on the People's Pharmacy, insights to bring you up to speed on COVID-19.
In the People's Pharmacy Health Headlines, COVID-19 is now the leading cause of death in the United States. That's the conclusion of an article published in JAMA. The authors note that the daily death toll from the coronavirus is roughly equivalent to the number of people who died during the 9/11 attacks. The number of people who die daily from COVID-19 is about the same as having 15 Airbus 320 jetliners crash every day. If current trends continue, COVID deaths will exceed daily mortality rates for heart disease and cancer. The authors note that the prospect of a vaccine offers hope for 2021, but that solution will not come soon enough to avoid catastrophic increases in COVID-19-related hospitalizations and deaths.
British public health authorities are concerned that a new variant of COVID-19 is highly contagious. The new strain is thought to be 70% more likely to spread than the older coronavirus version. Scientists have found that the new variant has 17 mutations, primarily in the part of the gene that codes for the spike protein. As a result, the government is calling for stricter lockdowns. Prime Minister Boris Johnson had hoped to avoid that over the Christmas holiday. However, infections in London doubled in just a week, with the highly transmissible new variant accounting for more than 60% of cases.
Sweden took a different approach to COVID-19 from its Nordic neighbors. The Swedish Public Health Agency decided last spring that it did not need to close schools, restrict hours in restaurants and bars, or encourage people to wear masks. The state epidemiologist Anders Tegnell told his countrymen that this approach would reduce the likelihood of a fall surge in COVID cases. He was wrong. Sweden is experiencing a staggering surge in cases that is overwhelming hospitals in Stockholm. Sweden's king, Carl XVI Gustaf, has admitted that the country has failed. He said the Swedish people have suffered enormously in difficult conditions. More than 8,000 Swedes have died. In comparison, Norway and Finland both have death tolls under 500. The government is now encouraging citizens to wear masks on public transportation. Non-essential workers are urged to stay home.
Now that the Pfizer, BioNTech, and Moderna vaccines are being shipped all across the country, tens of thousands of Americans are rolling up their sleeves. By now, most people are prepared for the common reactions such as pain and redness at the injection site, fatigue and fever the next day, and headache, muscle, or joint pain. A rare but worrisome side effect is also possible. There are a number of reports of anaphylactic allergic reactions. The CDC recommends that everyone receiving the vaccine wait at least 15 minutes after getting the shot to make sure that they're not reacting. People who have a history of severe allergic reactions should wait at least 30 minutes. Before anyone gets a COVID-19 shot, they should verify that the healthcare providers administering the vaccine have access to epinephrine and antihistamines in the event of a bad reaction.
Do you remember hearing that COVID-19 was no worse than the flu? A new study from France compares hospitalizations and deaths for both diseases. The researchers found that during a 2-month period in 2020, twice as many patients were hospitalized with COVID-19. That's compared to people hospitalized with influenza during the 3-month flu season of 2019. More worrisome, the death rate for hospitalized patients with COVID-19 was nearly triple that for influenza. In addition, ICU stays were nearly twice as long, and patients with the coronavirus were more likely to require mechanical ventilation.
Public health authorities worried about the possibility that Americans could be faced with a twindemic of influenza plus COVID-19 this winter. So far, though, the flu seems to be far less common than usual. Precautions to slow the spread of the coronavirus may be largely responsible for the lower burden of flu. This pattern is similar to that seen in the southern hemisphere between May and October. That's winter in Australia and other places below the equator. Their 2020 flu season was exceptionally mild. Let's hope it holds here, too. And that's the health news from the People's Pharmacy this week. Welcome to the People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. As we come to the close of 2020, the coronavirus pandemic is still ravaging the world. However, there are now 2 vaccines being administered in the U.S. We've turned to the coronavirus hunter for an update on where we are and what to expect.
Our guest today is Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology of the University of North Carolina at Chapel Hill. In addition, he's a World Technology Award finalist and a Fellow of the American Association for Microbiology. For the past 3 decades, Dr. Baric has warned that the emerging coronaviruses represent a significant and ongoing global health threat. We talked to him just before the Moderna vaccine got emergency use authorization.
Welcome back to the People's Pharmacy, Dr. Ralph Baric.
It's a pleasure to be here again. I hope you both are doing well.
We are, thank you.
And we hope you are doing well as well. These are challenging times. It's pretty astonishing how many people have been hospitalized, how many people have died, and how many people have caught this virus that you have been studying. Is it really now over 30 years?
Well, I've been studying coronaviruses for over 30 years.
Right, not this virus because it's new. Dr. Baric, vaccines from Pfizer and BioNTech and now Moderna and possibly soon AstraZeneca and Johnson & Johnson will be distributed around the world. What's your perspective? What's the latest information, and what should we know about these vaccines?
Sure. There's one vaccine already approved for use in the American population, a second vaccine that is under emergency use review and will probably be available for the U.S. population shortly. These are both messenger RNA-based vaccines that are very similar and induce very similar immune responses in vaccinees. Both vaccines, at least from the current status of the data, are about 95% effective. In other words, 95% of the individuals who receive the vaccine are protected from infection. Of the 5% or so that do get infected, they have, in general, very mild disease symptoms. And so these are fabulous vaccines, and I highly recommend the American public getting them.
Everybody says this is way better than expected. Why do you think these vaccines have performed so well? And what about the other vaccines that are on the runway?
So these are both messenger RNA-based vaccines, which are completely new vaccine technologies that have been under development for some time. And in animal models of human disease, both small and large animal models, they induce very, very robust antibody responses in vaccine recipients, including very, very high neutralizing titers. And neutralizing antibodies are those antibodies that are present in your circulation and tissues that bind to the virus and destroy its infectivity. And so they're very, very good at protecting from infection. The other vaccines on the market include a bunch of what are called adenovirus vectored vaccines, like AstraZeneca. These are adenovirus particles, which are normal human pathogens. They're mild respiratory pathogens that have had all of their pathogenic machinery gutted from the virus. And they've been engineered to deliver foreign transgenes, in this case, the SARS-2 spike glycoprotein. They also elicit fairly good antibody responses in vaccinees. And the AstraZeneca data indicates that these vaccines have about 65% efficacy in terms of ability to protect from infection. This is significantly higher also than what the standard had been set for any of these vaccines to achieve 50% efficacy. So although the AstraZeneca vaccine is not quite to the level of the mRNA-based vaccines, it is still very robust. In addition, there's a Janssen adenovirus vaccine, which we have not seen the phase 3 trial data on as of yet, because I think it's just starting phase 3 trials. And then there are two recombinant protein vaccines, one from Sanofi and the other from a company that's escaping me right now. And that the preclinical data in animal models and the phase 1, phase 2 studies in humans look pretty interesting. I think those vaccines will most likely, the latter three vaccines that I just mentioned, will most likely be reviewed in January, February, March timeframe.
Dr. Barrick, it is going to take a little while, or maybe longer than a little while, to really roll these vaccines out so that a lot of people get vaccinated. But I think people have the idea that, well, once I'm vaccinated, I don't have to maintain social distance. I don't have to wear my mask. Is that a misperception?
We know a lot about how these vaccines perform, but there are some things that we don't know about very well. Many of the vaccines that I mentioned induce a very strong protective immune response that works in your lower lung. And that's where all the severe disease is. Life-threatening disease occurs in your lung, and especially in the lower lung. And these vaccines completely protect against that. However, virus transmission occurs from infections that occur primarily in the upper respiratory tract, in the nose and the oropharynx region. And those are compartmentally quite different from the rest of the lung. And immunity is not quite as effective in those compartments as they are in the deep lung. And so it is possible that some of these vaccines may induce protective immunity, but allow for low-level infection in the upper respiratory tract and in your nose, and resulting in essence, the sniffles, or very mild common type cold events that cause you to sneeze. And you could potentially transmit to people who have not been vaccinated. So at this point, as you might expect, that kind of information is really scant, because there's only been about 15 to 20,000 people vaccinated with these two Moderna-based vaccines. We know that at the highest dose in animals, they induce a good response in the nose of these animals, but at a slightly lower dose, they don't. And because humans are so outbred and heterogeneous, it's quite possible that some individuals won't mount a really good response that protects from infection in the nose, and they could transmit to people around them. And we won't know that data until we study what happens in vaccinees for a longer period of time.
So, Dr. Barrick, it seems as if the long-term perspective is positive. Once we achieve whatever we mean by herd immunity, which might be 6 months or 9 months or a year away, we should be in pretty good shape. But right now, what we seem to have a short supply of is what I would call antiviral treatments. We know about the antibody treatments that the president received and his attorney, but they're not widely available. So warp speed has been great for vaccines, but what about right now for treatment? Things like EID 2801, which is a drug that you've been involved with the early research on, ivermectin, and in fact, the antibody treatments, which seem to be highly effective. Can you give us some insight? Because some of these treatments might be able to stop infections pretty quickly.
So what's available right now is remdesivir, which has to be given by IV, and that's its weakness. And the reason that that's a weakness has to do with the pathophysiology of COVID-19 infections. So when people get infected with COVID-19, they have very high virus titers early on, and then those titers start to fall off. As the titers start to achieve peak titer or fall off, you start having patients hospitalized with more serious disease. And then even as the virus then continues to wane in terms of its titer in an individual, the disease severity is going up because of immune pathology, the immune response to infection. So consequently, remdesivir is oftentimes given later in infection than it should, just because you have to be hospitalized to receive the IV administration of the drug. And so peak titers have oftentimes already occurred and the virus titers are already coming down. So it's less effective as it could be in terms of how it was originally designed, which was as a direct acting antiviral that attacks that initial high titer phase of infection. An oral formulation of remdesivir would certainly be much more effective in that as soon as people are diagnosed early in infection before titers peak, if that drug can be given as an oral administration, then that would be much more effective. And that's the promise of EIDD-2801. It's also a direct-acting antiviral. It will probably experience the same phenotype in that it's given later in infection. It will be less effective because it's not necessarily the virus killing you anymore. It's the host response to the virus that's causing death. But EIDD-2801 as an oral administration would be able to be given early. Human monoclonals have that same phenotype. These are also considered direct-acting antivirals. If they're given late in infection, they fail. So the Eli Lilly work demonstrated that if you give these monoclonal antibodies to people who've been hospitalized and are developing serious disease, the antibodies are not effective. But if they're given early again, where they can actually knock down virus titers, then they become much more effective. And the beauty of the monoclonal antibody is that it can be given as a single IV administration as compared to remdesivir, which is 5 consecutive days. So that's much more usable. The antibodies can be delivered more easily in a clinical setting as compared to the admittance into the hospital.
You're listening to Dr. Ralph Barrick, the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. He's helped make UNC at Chapel Hill a world leader in coronavirus research.
After the break, we'll talk about evidence that the virus started moving around the world before last December.
Some strains of SARS-2 have new mutations. Will that affect vaccine efficacy?
Some people have advocated for herd immunity. Is it a realistic goal?
Will we need new vaccines every year or so, like the flu shot?
If someone already had COVID-19, should they be vaccinated?
You're listening to The People's Pharmacy with Joe and Terry Graydon. The People's Pharmacy podcast is supported in part by CocoVia Memory Plus. CocoVia cocoflavanols support both cardiovascular health and cognitive function by promoting healthy blood flow, transporting oxygen and nutrients to vital organs and muscles, including your heart and brain.
CocoVia Memory Plus has 750 milligrams of cocoflavanols, the plant-based nutrients from fresh cocoa that have been proven to help boost memory.
CocoVia Memory Plus is backed by 4 clinical trials that demonstrate improvement in 3 different aspects of memory: long-term memory, spatial memory, and word recall. The studies show improved brain function in just 8 weeks.
You can try the benefits of CocoVia Memory Plus with a 25% discount off your first month. Use the code PEOPLES25. To get the full benefits, take it daily for 8 weeks. Cocovia is offering People's Pharmacy podcast listeners a 10% discount on subscriptions. That code is Peoples10. Learn more at cocovia.com.
Welcome back to the People's Pharmacy. I'm Terry Graydon.
And I'm Joe Graydon.
The People's Pharmacy is brought to you in part by Cocovia Memory Plus, a cocoflavanol supplement backed by 4 clinical studies that show significant improvement in 3 different aspects of memory. More information at cocovia.com.
Also by Verazana, an analytical laboratory providing home health tests for hormones, gut health, and the microbiome. Now with an annual health club plan. Online at verisana.com/health-club.
Today we're bringing you up to date on the research regarding COVID-19. What do we know about the origins of the SARS-CoV-2 virus that's responsible for the pandemic? What are the implications of mutations in this virus? We're fortunate to have one of the world's leading experts on coronaviruses to help us answer these questions.
Our guest is Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. He is a Harvey Weaver Scholar from the National Multiple Sclerosis Society and an established investigator awardee from the American Heart Association. In addition, he is a World Technology Award finalist and a Fellow of the American Association for Microbiology.
Dr. Baric, the origin of this virus is still contentious. And in fact, we've seen some reports suggesting that the virus was perhaps already circulating around the world late in 2019, before anybody had named it or was looking for it. Do you have any insights into whether or not those reports are credible, and how would such a virus have been circulating undetected?
Well, to begin with, some of the data from the CDC and from the group in Italy looks quite compelling, suggesting that there were infections that are occurring as early as, October or maybe even September. The October data is better than the September data. There are some issues associated with that, because if you would anticipate early circulation of those viruses should have been associated with more explosive transmission and increased occurrence of disease in populations, and that didn't really seem to happen between the months of September and January, at least in the United States and in Italy. So there's some issues. There could be some issues associated with it, but right now the data is pretty compelling. I'll just note that it's an important caveat to consider in any kind of discussions you have on whether this virus may have been circulating much earlier, is that to date, to my knowledge, the Chinese have not done any seroepidemiology studies to ask questions about whether the virus was circulating in China much earlier than December, as they reported. So my personal opinion is that this is a virus that originated in China, was circulating in China. In fact, there were big, what were called flu epidemics occurring in other major cities in China in the months of October and November prior to the emergence and discovery of SARS-2. And so it's quite easy to imagine a scenario where SARS-2 was hiding underneath this surge of flu infections. Also, just to keep it in perspective, last 2019, 2020, especially the fall and the spring of 2020, and the fall of 2019, we had a fairly big flu year that year. And so any kind of new virus like SARS-2 would have been, in essence, able to camouflage its existence, because physicians and those who were looking at cases would have anticipated that was just part of the flu epidemic that was occurring. And so the virus, in essence, was able to hide. The other thing about SARS-2 is that a large number of infections are asymptomatic, but you can still spread. And so you can have a fair amount of spread occurring early on prior to detection of the virus. And these silent transmission networks are quite important in the life cycle of SARS-2.
So, Dr. Barrick, if I'm hearing you correctly, you are suggesting the possibility that SARS-2, COVID-19, as we now call it, was circulating in China well before January, before it was recognized, and may have even been circulating in the United States, in Italy, and in other European countries in the fall of 2019.
That's right. I think you have to, I don't remember the exact statistics for the number of travelers coming from China to the United States, but it's something like 45 or 50,000 a month. You probably should check that. I could be off, but it's a large number of people come from Southeast Asia to the United States and Europe and elsewhere in the world. So low-level transmissibility, for example, you could have strains that were not as efficiently transmitted in China that required additional rounds of human-to-human transmission before strains evolved the capacity to be efficiently transmitted. So again, things are still unclear in terms of the early events of infection. I think the earliest strains that we've ever sequenced were identified or isolated from patients in the first 10 days of December 2019. So any viruses that had preceded that, we don't know what their sequences are. So we don't know what they look like. And we can't make predictions about their biology.
Well, speaking of mutations, we've been hearing from the UK that there may be some mutations that have occurred in the southern part of the country that are much more infectious. And I'm wondering if you can share with us anything that you've learned about the virus, its mutations, and what we might expect for the future.
I've certainly seen there. So to put this in context, the first really novel mutation that emerged that had a big impact on the biology of SARS-2 was a mutation that occurred in Southern Europe around late February 2019. And it was a mutation in the spike glycoprotein. It's called D614G. It's just a mutation to a glycine in the spike. That virus is much more transmissible. Based on studies in humans, it replicates to higher titers in the upper respiratory tract based on studies using primary cells from the nose and upper respiratory tract. And that virus is much more transmissible between hamsters. And the epidemiology suggests it's much more transmissible than the contemporary isolate that came out of China. These strains now are the predominant strains globally. So any kind of other mutations that emerge are emerging on top of these initial mutations that enhance transmissibility. Now, the English, the British are reporting a new strain associated with a large cluster of cases in England. I have not been able to find the sequences yet from those strains, although, quite frankly, this was reported, a day and a half ago. And I looked yesterday, but I didn't have a chance to look today to see if they've published the sequences, the mutations associated with this phenotype. I would probably argue that it's still early. They could easily be right, but it's still early. If this is a much more transmissible variant than the D614G, we would expect that it would replace that as it spread throughout the country and that it would appear simultaneously on other continents and begin to displace the current circulating strain on those continents as well, just like what happened back in February. And in fact, the D614G variant, when it emerged in late February, by the end of May, it was the dominant form globally. So we'll know shortly whether this truly is a more transmissible variant and whether it alters the immunogenicity in any way that might be a concern for vaccinees.
Now, exactly. That's the question. Will the vaccines, the various vaccines, be able still to protect people even after the virus has mutated?
Well, that's a good question. So to put this in context, the D614G variant from February 2019 actually that mutation made the virus more susceptible to neutralization as compared to the ancestral strain. And it makes sense, right? Because almost everyone on the planet is naive. That means they have no existing immunity against this virus. So there's no selection against in terms of neutralization, or the virus doesn't need to protect itself from people who had been exposed previously. So it modified its spike-like protein so that it would maximize transmissibility. I would anticipate, given that only 5 to 10% of the people on the planet have been infected with this virus, there's still plenty of naives around. They're in a massive excess. So we could have additional mutations occurring on top of this mutation to even further enhance transmissibility. Would it make it more resistant to neutralization or less resistant to neutralization? We don't know. As soon as the mutation is reported, we will have people in many laboratories around the world ask that question and actually use antibodies from vaccinated individuals to see how well those vaccines, whether they induced a strong protective response against the new variant. My guess is the answer will be yes, because there is not that much pre-existing immunity to select for strains that would be resistant to neutralization. That's my guess, but we'll see.
We will indeed. Dr. Barrick, you've just suggested that perhaps 5 to 10% of the population globally has now been exposed and presumably has at least some immunity to the virus. My question is, people have been talking about herd immunity. What will that take? Or is that even a realistic goal?
Well, there's 2 ways to achieve herd immunity. In general, what herd immunity means is that about 70% of the population has been exposed to the virus and has developed some level of pre-existing immunity against the virus. And that should protect them from reinfection. So that there are 2 ways to achieve herd immunity. The hard way, which is to let the virus run rampant in the population and cause 1% mortality rates as it makes sure it infects everyone on the planet. Let's see, there's 7 billion people on the planet. And my math puts that down to somewhere around 70 million people would die. And it's not really the way we want to achieve herd immunity on planet Earth. The better way to do it is through vaccines. And so in that case, the vaccines elicit a strong protective immune response, and that allows the population now to achieve herd immunity. Now, what are the factors that influence herd immunity? Well, people who become infected with SARS-2, in general, those who get more serious infections have better immune responses than those who don't. So if you have very mild infections, you may have a mild immune response that wanes over time and allows you to be reinfected in the future. And so that impacts herd immunity. The other things that impact herd immunity is the virus is now under pressure to figure out a way how to survive in the presence of all these hosts, which are immune. And the way influenza virus does it is it changes its surface. And so it escapes herd immunity and now can continue to infect people. In the vast majority of cases, infections a little bit less intense, but there can be variants that can cause severe epidemic waves of disease with a fairly high amount of mortality, especially in the elderly who don't have as robust an immune response as the younger individuals.
So this is why we need a brand new flu vaccine every year to try to adapt to that new surface protein.
That's correct. Every year, there are scientists all over the world who collect flu strains, and they all get together in January or February of the new year, and they try to decide which strains are they going to incorporate into the vaccine formulation for the fall. And it's a very, very tight timeline. They have to do estimates of which ones they think are going to cause the pandemic or the epidemic in the preceding year. And they choose. And sometimes they choose wisely. And the vaccine's great, and sometimes they don't.
Well, this whole issue of vaccines, Dr. Barrick, brings up the question of if someone has already had COVID-19, and there are many, many people who have, many of them asymptomatic or mild symptoms, should they get a vaccine? In other words, they've recovered from the virus, but should they get a vaccine, and where should they be in prioritization? Should they be high, medium, or low?
Well, those are great questions. And I think the immediate priority in the United States is to protect healthcare workers. We don't need to continue to lose healthcare workers that are working 18 hours a day to try to save people's lives, especially in the current scenario where the hospitals are completely either overrun or nearly overrun with cases. And so that's the immediate priority. Let's vaccinate them. The second priority are the elderly, especially those in retirement communities that capture, I don't exactly remember the percentage of deaths, but something like 40 or 50% of the deaths have occurred in the elderly in retirement communities and assisted care communities. And so we need to get them vaccinated as a priority. After that, individual states have to make recommendations of what they're going to do and who they're going to vaccinate. And the truth is, the United States have had cases of COVID-19 since January 20th, 2019. And we are beginning so we've had the opportunity to track what happens to the immune response in terms of the neutralizing antibody titers and the cellular immune responses for about 11 months now. And that data will be coming out shortly in terms of how robust it is, what the durability is, and is there any relationship between the magnitude of the response immediately after infection and the longevity of the response in terms of its likely protectives. And hopefully some of that data will guide the decisions about who should receive the vaccine. Certainly high-profile political figures are being discussed as who have been infected are being discussed as having that they should receive the vaccine, which would suggest that many people who have been infected will be given the vaccine. The question's priority. My priority would probably be to focus on those high-risk individuals who haven't been infected yet.
You're listening to Dr. Ralph Barrick. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. He's a Harvey Weaver Scholar from the National Multiple Sclerosis Society and an established investigator awardee from the American Heart Association. In addition, he's a World Technology Award finalist and a Fellow of the American Association for Microbiology. He's helped make UNC at Chapel Hill a world leader in coronavirus research.
After the break, Dr. Barrick offers his ideas on the future of this pandemic.
With the worst-case numbers, a lot more people will die in the United States. Even in the best case, another 200,000 people could die before the pandemic is under control.
Dr. Barrick has some suggestions for gathering safely with our families.
How can we prepare better for the next virus that may emerge?
We'll also find out how soon Dr. Barrick thinks we might see significant improvement in this pandemic.
You're listening to The People's Pharmacy with Joe and Terry Graydon. This podcast is brought to you by the Verrazana Health Club. This comprehensive home testing service enables you to track crucial health markers of gut health, inflammation, metabolism, hormones, thyroid function, and many other organ systems. Regular testing can help detect health imbalances before they lead to sickness. Online at Verrazana, verizana.com/health-club. Get 50% off the first month with the discount code PEOPLE50.
Welcome back to the People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon.
The People's Pharmacy is brought to you in part by CocoVia Memory Plus, a supplement with 750 milligrams of cocoflavanols, the plant-based nutrients from fresh cocoa that help support memory. More information at cocovia.com.
Also by Verizana, an analytical laboratory providing home health tests for hormones, gut health, and the microbiome, now with an annual health club plan. Online at verisana.com/health-club.
Today, we are seeking the latest insights on the progress of the vaccines and treatments for COVID-19. How soon could the Pfizer, BioNTech, and Moderna vaccines slow the spread of the virus?
Our guest is Dr. Ralph Barrick, the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill.
Dr. Barrick, many scientists predicted a second and even this third wave, which a lot of non-scientists, they didn't think it would ever happen. But now the crisis is worse than ever in the United States. What are your future predictions? What do you imagine will occur here in the U.S. and around the world going forward? Are we going to see a fourth or a fifth or a sixth wave?
Let me start off by saying that Americans live in an environment that's surrounded by science. There are incredible technologies at their fingertips every day that they access and use. We have incredible capabilities in terms of being able to monitor and change our environment and manipulate our environment for our comfort and convenience as human beings. Science has transcended the world for good and in some cases for bad. But every day, every single American is surrounded by the beneficial effects of scientific discovery. So it's amazing to me that people are so critical of the scientific community and their ability to predict events in the future. Scientists have been stating that we are going to see increasing waves of disease. Scientists have been saying that we know coronaviruses like to transmit, and they transmit more efficiently when it's cold in the winter than they do in the summer. And so the fact that we had so much transmission occurring in the summer made it pretty obvious to the scientific community that we were going to see a massive increase in cases come the cooler months starting around October, November. And that's what we've seen. The scientific community is telling us, despite the fact that we're rolling out vaccines, we are still looking at somewhere between 200 and 300,000 deaths before we begin to get a handle on this pandemic. And that's because of the time required to manufacture and roll out the vaccines.
Let me ask you, are you suggesting, in addition to the 300,000 deaths we have already, that there could be another 250 to 300,000 deaths?
Yeah, well, let's do the math. 3,000, about 2,500 to 3,000 people are dying every day in the United States. So that means 30 days from now, we will have 400,000 deaths. Even the best estimates of the vaccine put us at somewhere around 40 million doses, which means 20 million people get vaccinated by the end of January, pretty much. So that means we still have, can't do the math real fast in my head, but we're perhaps now at 20% herd immunity in the U.S. population. That means we will continue to see cases on the range of anywhere from 1,000 to 3,000 from January to February, and that will then progressively drop from February to March, March to April, April to May, until so many individuals in the United States get vaccinated, assuming again that 40% of the U.S. population doesn't pass on the vaccine, which means that if they do, we're going to have a lot more deaths than I just told you. But when you do that math, we're looking at 200,000 deaths, best case scenario. So that's my prediction. That's my prediction. The CDC, same thing. The NIH,
200,000 additional deaths to where we are now.
best case scenario.
Worst case?
Depends on the American public. If 40% of the American public decide not to get vaccinated because they're concerned about the potential 1 in 500,000 to 1 in a million adverse responses that might be associated with a vaccine versus a 1% mortality rate associated with infection, that decision will determine how bad it will be after about April or May.
Very sobering.
It is. A couple other factors. Number one, the other factor I think that's important to remember is that none of these vaccines have been tested in anyone under the age of, I think, of 16. And so I know that Pfizer and Moderna and several other companies are about to roll out clinical trials in younger children, and we need to have that information. So before we can begin to vaccinate younger children. So that's also going to cause a delay. But it's sobering. And this is this is in resource-rich nations like the United States and Europe. Think of how quickly vaccines are going to make it to resource-poor nations and the developing world.
Given what you've just told us, Dr. Barrick, what are your recommendations when it comes to family gatherings? How should people behave?
So number one, what's the fraction of the American public that's going to take the vaccine? I think we should all pray that it will be a high number. The second thing we can do, we wear masks because masks work. If two people are wearing masks and they're wearing them appropriately, there's about an 80% reduction in transmission. Just think of what an 80% reduction in transmission would mean in the United States today. We have 200,000 cases a day. An 80% reduction in that means we have 16,000. No, that is 40,000 cases. There would be 40,000 cases a day. That's a lot less mortality. That's a lot less stress on the hospital system. That's a lot less risk for every single person walking around the streets of America. So wear masks. It's a simple solution and it works.
Dr. Barrick, what about next time? The next virus that emerges, should we be better prepared? How can we prepare better?
Well, I think, number one, I want to at least herald the triumph of science. We have never rolled out a vaccine 11 months after a new virus in a virus family that we had never created a vaccine for previously. That is 95% effective. That's an incredible accomplishment of the scientific community. And people need to realize that doesn't happen by accident. These messenger RNA-based vaccines were being studied before the COVID-19 pandemic. They were being developed for use with MERS coronavirus, which occurs pretty much only in Kingdom of Saudi Arabia. Our group worked with the VRC at the NIH to test these vaccines in animal models of MERS coronavirus pathogenesis, and we knew they worked. That was one of the reasons why the mRNA-based vaccines were supported for Operation Warp Speed, because of their potential to control not only MERS infections, but also flu infections. So there was a lot of basic science that had been supported by the American public through the National Institute of Health and through support by both parties in Congress, have always been very, very supportive of the National Institute of Health and basic science that could be translated into clinical products that would make a difference in people's lives. And we're getting the benefit of that today. The second thing is there had been a fair amount of research on trying to identify broad-based inhibitors against coronaviruses, done almost exclusively in two labs in the world. That was Mark Dennison's lab at Vanderbilt and my lab at UNC, in collaboration with Gilead and a few other groups. But we were really trying to identify a drug that would work against all coronaviruses so that it could be applied. We'd identified remdesivir. And that's why it was used in the first patient, because there was a good body of literature that said it could be used. If we'd had another 10 months to a year working with Gilead, we would have had an oral form of that drug, which would have saved many, many more lives than the drug already saved. It would have been much better. A delay of 10 months would have probably allowed for 2801 to be ready as an orally available drug for all coronaviruses. So the good news, I guess, is that for coronaviruses, we will probably have orally available drugs that will likely succeed in future coronavirus pandemic settings based on remdesivir and 2801. We will have therapeutic human monoclonal antibodies that will work against all SARS-related viruses that exist in nature that we are able to culture in the laboratory. That doesn't mean that, to put it in perspective, there are literally millions of variants of SARS-related viruses, of which we can probe about 10. But those 10 are carefully chosen for maximal antigenic variation. So the idea is if it works on all the bookends of the SARS-related virus family, that it would work against the unknown that might cause a future pandemic. And so we'll have a monoclonal antibody that will be on the shelf that could be used. And then finally, we have vaccine platforms that we know work against coronaviruses, and we're going to have good immunogenicity data about how people respond, what the magnitude, the durability, and the waning of the response might be, and how well they protect against related and distant strains. So I think we're going to be much better off in the future if it's a coronavirus, if it's a SARS coronavirus, if it's a MERS-like coronavirus. We'll have drugs, but we won't have antibodies. We won't have a vaccine. So, we're looking at 5 to 10 months instead of 10 months.
Dr. Barrick, I wonder if you can take a look into your crystal ball and give us some idea of what we should expect the rest of this COVID-19 pandemic to look like. How soon can we expect things to start getting better?
I can tell you, I'll tell you what I think. I want to preview that comment with the statement that there are lots of people that argue with me. Okay. So
sure.
So my gut feeling is that in the United States, the vaccine will be rolled out effectively, and that we'll be seeing the back end of this pandemic by August, September of 2021. Now, that doesn't mean that it's going to go away. It means that this sort of explosive pandemic paralyzing event that we're living in right today will be over for the most part. What won't be over is that we will still have infections occur. We won't have massive pandemics because the number of susceptible hosts are limited. So instead of one person transmitting into 15 people in some sort of setting like a restaurant or a bar, they may only transmit to 2 or 3. And so that would result in a significant reduction in the RO, or the replicative rate, sort of the transmission rate of the virus in populations, and result in less cases. And again, depending on the percentage of the American public that is willing to get vaccinated, the number of cases and deaths will be, painful, but not paralyzing like it is today. It may be more along the line of a flu season, where you have 15 to 20,000 deaths in the preceding fall and winter of 2021, 2022. Now, for the rest of the world, it's a different scenario. Currently, most of the wealthy nations in the world are buying the vaccine doses. And so the current vaccines, the mRNA-based vaccines, require critical cold chain elements that make them difficult to be able to distribute in most locations in the world. Even in parts of the United States, it's going to be difficult to get the Pfizer vaccine to, just because of the cold chain requirements. So the key thing for the rest of the world are these recombinant protein vaccines that are currently entering phase 3 testing. Will be complete sometime soon. If those recombinant protein-based vaccines look effective, then they can get rolled out to the rest of the world. But I think that's going to be slower. Obviously, it's a lot more people. Obviously, the logistics of delivery are much more complex. And so this pandemic is going to be still raging in parts of the world through 2021 into 2022, which means that variants are going to be continually reintroduced into the United States. So again, depending on our public health practice and our willingness to get vaccines, we'll either be protected from them or we'll have localized outbreaks of disease that occur in the people that are not immune. And that's dangerous. And it's dangerous because that allows variants. When you have a mixed population, some with immunity, some without, you're putting in a scenario where you are maximizing the possibility of selecting for escape mutants to the immune response that has been elicited in vaccinees and infected individuals. And coronaviruses do have a history of antigenic variation. They do it a little bit different than flu, although they do use the flu-based approach by mutation. But they can also delete part of their spike glycoprotein and result in new tissue tropisms in terms of where they target in the host and their ability to escape vaccines. And so we don't want to give the virus a lot of time to try to figure out how to get around immunity.
Dr. Ralph Baric, thank you so very much for bringing us up to speed on the SARS-CoV-2 virus on the People's Pharmacy today.
It's a pleasure being here again today, and you all be safe over the holidays and take care. Pleasure talking with you.
You've been listening to Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology of the University of North Carolina at Chapel Hill. He's a Harvey Weaver Scholar from the National Multiple Sclerosis Society and an established investigator awardee from the American Heart Association. In addition, he's a World Technology Award finalist and a Fellow of the American Association for Microbiology. He's helped make the University of North Carolina at Chapel Hill a world leader in coronavirus research.
Lynn Siegel produced today's show. Al Wodarski engineered. Dave Graydon edits our interviews. B.J. Liederman composed our theme music.
The People's Pharmacy is a co-production of North Carolina Public Radio, WUNC.
The People's Pharmacy is brought to you in part by Verizana, an analytical laboratory providing home health tests for hormones, gut health, and the microbiome, now with an annual health club plan. Online at verisana.com/health-club.
And by CocoVia, offering plant-based nutrients in the form of cocoflavanols for brain and heart health. Online at cocovia.com.
To buy a CD of today's show or any other People's Pharmacy episode, you can call 800-732-2334. Today's show is number 1241. The number again, 800-732-2334, online at peoplespharmacy.com.
When you visit the website, you can share your thoughts about today's show.
You can subscribe to our podcast through your favorite podcast provider. We post it on our website every Monday morning.
At the website, you can sign up for our free online newsletter to get the latest news about COVID-19 and other important health stories.
In Durham, North Carolina, I'm Joe Graydon.
And I'm Terry Graydon. Thank you for listening. Please join us again next week. Thank you for listening to the People's Pharmacy Podcast. It's an honor and a pleasure to bring you our award-winning program week in and week out. But producing and distributing this show as a free podcast takes time and costs money.
If you like what we do and you'd like to help us continue to produce high-quality, independent healthcare journalism, please consider chipping in.
All you have to do is go to peoplespharmacy.com/donate.
Whether it's just one time or a monthly donation, you can be part of the team that makes this show possible. Thank you for your continued loyalty and support. We couldn't make our show without you.